Thank you so much for the opportunity to speak today to the MEF2C community. Thank you to Anne and the organizers. My name is Misha, and I'm a genetic counselor in the Simon Searchlight study. Today, I'll be structuring my talk with some general information at the very beginning, and I'll be diving deeper and deeper with examples of single nucleotide variants, copy number variants, and in the end, cover some general information about reading your genetics lab report. So genes are, the MEF2C genes are like recipes. This is what I think of when I think of genes. They are important instructions that are intentionally made that way to make a particular protein. So the MEF2C gene is important for the MEF2C protein. And I think of the protein as the product, the baked good, if you will. The gene's recipe needs to be followed in a really particular way so that the outcome, the product, is functional and the product you need. And so if you have too much sugar or not enough water in a recipe, it will end up messing up that product. It's the same way in a protein that if you have certain variations to that instruction, then it can ultimately disrupt the protein in some way. And proteins in this context are not actually proteins you eat. So I hope the example is not misleading. But proteins in this context are not the proteins you eat, but the proteins that are the building blocks of life. So in this presentation, I'm meaning proteins that are like enzymes, hormones, important for antibodies. It's important, for example, if you have a wound or you've cut yourself and your body will heal. Those are active proteins within the cells that will come together to help heal that wound. And so proteins are really the building blocks of functional life. Additionally, it's important to cover that genetic variations happen within every single one of us. I have genetic variants that are different than my parents, and they have genetic variations that are different than their parents. This happens in every single pregnancy. There is cellular machinery that is copying the genetic information to make sperm, to make eggs, and there are mistakes that happens, and that is a common occurrence literally in every single pregnancy. It's only when these genetic variations ultimately affect the proteins that how they function or where they're localized, or get rid of the protein entirely, that this can cause developmental or health challenges for a person. So if it's a problematic variation. But in general, most of us have these variations, and the most genetic variation that happens is actually not problematic to us in general. So now as we dive into an example, DNA, it's important to cover that DNA has 4 different letter codes. So you likely have seen this as well in your genetic lab report and the other talks that might have happened today, that there are 4 letter codes for DNA. So it is either A, T, G, or C. Within a gene, you'll have these letters in a particular order that read out and make this protein. And I'll dive more into that information. But let's dive into different examples of genetic variants. So single nucleotide variants can result in many different types of genetic variations. So one possible example of a single nucleotide variation is a missense variant, a single letter change that results in a protein amino acid change. And again, I'll dig deeper into this, but for now, what you can see is that in this MEF2C variation, and the C dot always means your DNA, the DNA code, and the P dot always means the protein code. But in the gene region of MEF2C, usually at site 113, there's a T. But in this person being sequenced, there's a C instead. And this single letter code means that instead of the protein usually having a leucine at site 38 of this resulting protein, there's a proline. So in this example, this would be it's expected in general that a protein would be produced, but the protein is altered in some way. The resulting function, so what happens when this amino acid is changed, is different in every single genetic community. So experts in your community would have a better understanding how the protein is altered when there is a proline instead of a leucine at this site. But this would be an example of a missense variant. A single nucleotide variant can also cause a nonsense variant. So in this particular case, we have a MEF2C genetic change at 860, where in the reference genome, so where the lab is using a comparison sequence of MEF2C, a DNA reference, that usually people have a C at this site. This person being sequenced has an A. This does not result in this case in a missense variant, but actually a nonsense variant. And I'll dig more into this as well. But here you can see that how this is represented is that normally at site 287 of the protein, there's a serine, but here this little asterisk, and it might show up on a genetics lab report as a TER or termination, or it will show up on a genetics lab report as an X. Those are all the same to say that this is a nonsense variant and this is a stop, that a stop was introduced in that protein. So another way to look at this type of or to call this type of protein variation is a protein truncating variant. So now I'm going to walk through more detail of how it goes from DNA to the protein. So to do that, you have to think of this intermediate molecule. So when there is a DNA sequence, a genetic sequence, a gene sequence, I mean, it's not that that recipe is cut out of the DNA and used once. No, just like a recipe card that you'll have that you'll come back to many, many, many times again and again. What you need is to read the DNA. So there's cellular machinery, as mentioned previously, that reads the DNA and it makes an intermediate molecule. It reads off the DNA and it makes what's called an RNA molecule. This is an intermediary molecule before making and translating, and that's the word in genetics, that going from RNA to protein is a translation. But there's an intermediate RNA molecule. And in the scientific world, it's actually very well known how this happens. And how this happens is that actually the RNA is read as a three-letter code. This you can think of as a three-letter word in a sentence. And so it's read as the cat sat, for example, in this three-letter sentence to make the protein come together. So in this example, let's walk through, in this particular molecule, you have a GCG. And the research and scientific community have broken down and created this rubric, this chart. It might look like a circle. It might look like it can come in different forms, but essentially they're all saying the exact same thing. Here is how the cell reads these three-letter words in order to make the protein. And every single gene has a start, intermediate amino acid words, and a stop. In general, a general gene, that's how we can make genetic databases for humans, we have genetic databases for mice, where we can say, here are all the genes we're finding in the sequencing. We're not sure what all of these genes do. There's so many genes within every living organism, and researchers dig into and research what these genes might do, but we can tell what a gene is because we already know how to read the DNA. And so there's always a start and there are stops. So that's important, and we'll cover that in the next example. But if you look at how to read this chart, we have this first letter word where it's G, C, G. So first position G, C, G. That gives you this alanine, this A in this example. So I'm not going to walk through the other ones, but what you do is you look for the first position, you find the second position in this category, and then you find the third position from this other side, and it leads you towards which amino acid would be read by the cell. So just highlighting again that there are three-letter words for stop. There are three-letter words to stop the production of a gene. And if a genetic variation introduces letters or shifts the letters in a way, it can result in a premature stop. A stop before the gene should have had one, or a protein truncating variant, as we discussed. So, more examples. This is a different type of single nucleotide variant. This is also resulting in a nonsense variant, but what happened in this case is there's a single DNA deletion of a letter, and that shifts the frame. That shifts the three-letter word reading frame. So in the usual code, in the reference code that's being used in the lab, you would see in most of us in this region of MEF2C, AACT. But what's happened in the persons and the participants sample that we're using as an example here, this second A was deleted. It was removed. This is the genetic variation that happened in this person. And now the three-letter word is ACT in the DNA. What that results in the RNA code, and now I have to introduce a little more complex information, that the RNA made off this DNA, has another letter that it uses. So DNA is, A is always matched with T, and G is always matched with C, but RNA works a little bit differently. Whenever there's an A in the DNA, in the DNA code, a U will be placed. So A and U in the RNA. Whenever there's a G or a C, it will be, if it's a C, there'll be a G added in the RNA. If it's a G, it'll be a C added in the RNA. They're interchangeable. And whenever there's a T in the DNA code, there'll be an A placed. So as you can see, I've done the work of the transcription here, but going from DNA to RNA, this is the usual expected code. It would be UUGA, but this participant's mRNA would look UGA. So if you were to follow that, usually UUG would be UUG, would be leucine. But what happened with this particular single nucleotide variation is a UGA. So you can see still a U, but now we're over here, GA, which results in a stop codon, which results in a stop. So this is a protein truncating variant, but this is a single letter deletion. But we would still call this type of variation a nonsense variant. You could sometimes call it a loss of function variant. It's assumed that this type of variation would result in a loss of the protein function. Now there's another type of example that you can have with a single nucleotide change. I'm not going through exhaustive examples, but I'm just trying to showcase that there are so many different ways that the same type of genetic variation can result in different type of variants. And it always is very important to assess the variant and the variant in its particular context. So here we have now a frame shift variant. This is also a result from a single letter deletion. So here you have in the usual code a GAC, but the person is missing an A in this case. And reminding you of the DNA to RNA, I've done the work of adding in the participants' extended code as well as their mRNA code, just to show that In this case, the way that the genetic variant shifts the frame is an important feature of this type of genetic variation. So, what has happened when there was this letter removal is that there's an insertion of several more amino acids, and they give you the number here. It's 8 amino acids were inserted that were or now included that weren't normally the amino acids that should be there. So it shifts the frame. And then at the end of that 8, there's a nonsense variant. So again, you're still seeing that stop codon, that nonsense being introduced here. And I'll show you very specifically the amino acids that are now there. So normally a person will have a leucine at this site, and that's indicated here. The lab is saying that normally there's a leucine, which if you look at the chart, it would be a leucine. But what happens now with the shifted code is that you have these particular amino acids, and at the end of the 8th one, there's a stop included. So I've done the work. We won't walk through every single amino acid, but if you were to pause and look, you could see that every single there's all these 8 new amino acids, and then a stop is introduced. So you could call this type, and we would usually call this a frame shift variant. It does have a nonsense, a stop codon that is introduced, and it's also a protein truncating variant. It would also be assumed, although there's always exceptions in genetics, and there are general rules, but we always look at every single particular context for each person individually. But it would be assumed that this would be also a loss of function variant. So now to look at some copy number variants. So copy number variants, meaning that there is a large, and I'm using quotations because it's a loose description. I'll get more specific on the next slide, but a large amount of extra or missing genetic material, amount of genetic material on a chromosome. So this is just a cartoon of a chromosome, an example of where there might be a break. In a deletion context, it would make the chromosome shorter. In a duplication context, it would make the chromosome longer. More specifically, when we're wondering about a deletion, what would be considered a deletion variant, going by the scientific definition, it's defined by 50 base pairs to several thousands. So what that means is a deletion variant, when we think of one nucleotide being deleted, we would usually look at the protein consequence and call the variant by its protein, like we're saying, frame shift or nonsense. But when we think of a deletion variant or a copy number variant, it's 50 of those nucleotides in the DNA or more. And to dig deeper, if you were to have a copy number variant, you might have seen a particular number. So this is the cytogenetic location of the MEF2C gene. I'll go through what this means to say that this is an example of the chromosome 5. There is a P arm of every single chromosome. It's given to the arm of the chromosome that is shorter. So you can think of it as the petite, the small arm. And the Q arm is usually whichever arm of the chromosome is longer. The region where MEF2C is on the Q, the 5Q, you can see it here. And then this banding pattern represents how the chromosome is labeled. If you go to get a specific type of genetic testing and there's chromosome labeling, this would be a karyotype, then there's a specific chemical that's used that causes banding, and the banding is very characteristic for every single chromosome. It's how this long string of DNA is compacted into the structure so that it is really small and fits in our cells pretty easily. But this way that it's compacted is very patterned, and the banding so you can see the band numbers. They increase, or they have a particular way that they are named. And so MEF2C is on this 5Q region 14.3 on chromosome 5. And to go further, Whenever you get a result for a copy number variant, you will see these elements, these pieces of information on your genetic lab report. You'll see the reference. So this in orange is the reference that the lab used. The reference genetic information that each lab uses. And there's widely used ones, like this is a common one that's used, but it is updated regularly every few years, every 5 years or so. But as we sequence more and more people in general worldwide, we learn that what genetic variations are found in the general population and what are not. So as we're finding more and more genetic variation in the population, it's important to include that in the reference genome to say, here's what normal variation is, or here's what regular variation is. So every time you have these breakpoints, you have to look at it with the context of the reference genome that the lab used for their reference. So they will include that on the genetics lab report. You'll have the cytogenetic band information, as we said, what region is the gene on, and you'll be given the genomic coordinates. You'll be told here is the first breakpoint end, here's the other breakpoint end. The last piece that you'll see is the information of, is it a deletion? Is it a duplication? So ×1 means that you only have one, this person only has one copy of this genetic region. And so that means they don't have two, which is the typical, most of us have two copies of genetic information, from one from our mom, one from our dad. So on a genetics lab report, you actually won't see for the 5 chromosome, you won't see ×2 because any regular variation isn't being reported to you. But you will see ×3 if there's extra information on the 5 chromosome that might be problematic, or ×1, which is a deletion. So this is all very important information for copy number variants. Finally, there was a question about how big is the MEF2C gene. So the MEF2C gene is over 160,000 base pairs. That includes this entire region of the gene. But this region of the gene within our DNA, within our genome, only has a subset of parts that are important for the protein coding. So here, if you go to the source I'm using is the UCSC genome browser, it's widely used but publicly available and free. If you were to go here, you could find the MEF2C gene, and the line across shows this region of the DNA. This is where the MEF2C is. But the vertical lines here are the regions of where the DNA is important for protein production. These pieces in between may or may not be important, but we would think of them as the introns, or the in-between DNA that's holding the pieces of important information together. So when cellular machinery comes to make the RNA is made off of these particular vertical lines, and then the in-between is cut out. And so this is just the MEF2C gene region. So now I feel like I might be running out of time, but we'll touch briefly upon an example genetics lab report. So this is a cartoon example. It's written out. Not every genetic lab report will have the same pieces of information, but they often will have the items that I have itemized here. The layout also is very lab dependent, but in general, the results are presented on the first page of the potentially 9-10 page genetic lab report. Often your clinician, the evaluating provider, will provide some indication to the lab to gear their genetic sequencing. What type of genetic variation are we looking for? So they'll give some indications for testing. And then when the lab provides results, they will list the findings in often something like this way. So the result in this particular example is that this person has a MEF2C finding, and the lab is listing the reference that they used. So in this case, because it's a single nucleotide variation, they need the transcript information. So here is the transcript that was used that in order to look and reference the person's genetic variation. Often they will include to help the providers give some background information. So that's the next two pieces here. Because as we mentioned, there's more than 20,000 genes. It might not be that a provider knows every single genetic condition that's a possible result. But the lab will provide some background information. So is this gene characterized to be associated with a developmental condition or a genetic condition? Yes or no? So is it a candidate gene, or is it uncharacterized, or is it characterized? We know that MEF2C is associated with a neurodevelopmental condition. Often they will include the OMIM name. So this is the OMIM. This is a online medical resource that clinicians and medical providers use widely, and genetic lab reports will also list any OMIM information on the genetic lab report. I've included that here. This is the reference number, and this is the name that's listed under the OMIM category for MEF2C. This piece is also more background information, usually for a provider, but also, of course, understanding the genetic condition. It's the mode of inheritance. It's to say, when there is a problematic genetic finding in this genetic condition, how does it present? The mode of inheritance in this case is not indicating how was this inherited. Sometimes I feel like that language can be a little misleading, but it is considered MEF2C as an autosomal dominant condition. So when there is one problematic genetic variation in this gene, then this does result often with a developmental or a genetic condition. Another type of mode of inheritance, for example, is autosomal recessive. So in those, you might have heard of cystic fibrosis, for example. You need two problematic variations in order to have the genetic condition. And so that mode of inheritance would be autosomal recessive. Where we start to get into the actual participant or person information is often in the next few pieces of information. So the genotype, how was this variant found? Was it homogeneous? Was there two copies? Or often in the case with MEF2C, you'll see one copy, you'll see heterogeneous. So now it's information that this person, they found one copy of this genetic variation, and it was de novo. So in this example, there was likely parental testing. So that's the only way to determine if something or label something de novo. But was there a blood sample taken from mom and dad, and were they tested negative? And so in this particular case, it was found de novo, arising new in that person. And what is the variation and the variant classification? So by now, you are likely familiar with looking at this. The C dot is the DNA letter change. So at site 43 in the DNA, usually there's a C. This person has a T. And it results in the protein change, the P dot of an arginine at amino acid 15 to cytosine. But now comes the important part of the variant classification. Is this problematic? Are we aware that it's associated with a neurodevelopmental condition? And yes, this is a pathogenic variation. Variant interpretation comes in three sort of main buckets of categories. So the benign or likely benign, those are the two subcategories in this bucket. But you wouldn't normally see benign or likely benign labeled on a genetics lab report, because any benign or any non-problematic findings, they're not really reporting in general in this context. And so this would indicate that there's a genetic variation, but we see it in a population that doesn't have a neurodevelopmental condition or a problem with that feature being assessed. So we're not considering it a problematic variation. Then we have this likely pathogenic and pathogenic. As mentioned, there is a high level of certainty that this genetic variant has a consequence to a person's development and the resulting protein's function. The most annoying variant, but it is utterly common, is the variant of uncertain significance. So this is in the case where there might be some evidence to say that it could be a problem, but it's not highly confident that it's causing a problem, and it might not be a problem. So it's a variant of uncertain significance, or a VUS, or a VUS, depending on a person's preference of talking about it, but they're all essentially the same thing, that more information is needed either over time, more scientific understanding, finding more people with a genetic condition that has the same genetic variant, or additional information like testing parents can sometimes help reclassify or help with the interpretation of a VOS. So the lab does interpretation, as you saw in the genetics lab report. There will be a level of interpretation. If, for example, a person has a VUS, and in general, the lab is often very cautious where they add something like this, where clinical correlation is recommended. So that means to say that the evaluating provider, the person doing the genetic testing, needs to also critically consider the person in front of them and the genetic findings that are there, in order to sometimes help reclassify a VUS, that your provider could do that additional testing for the parents and help with criteria to understand if this is more likely to be a problem or not. So there is also a layer of the provider's role in interpreting a genetic variant. Thank you so much for your time. That's the end of my Genetics 101. Please send any questions my way. I'm sorry that I can't be there live in person today. And then you can also follow us on many of our social media channels for updates and other types of research information on our Facebook, on X, on YouTube with our informational videos, or on LinkedIn.